I'm Kevin Kraus, CEO of Transformative Biotech, and I've spent more than 25 years in diagnostics and in personalized medicine, including with companies such as Ventana Medical Systems, Roche, and Genentech. And one of my passions is bringing innovations in science and medicine to patients and their providers. And that's why I'm so excited to talk to you today about the technologies that we have to simplify PCR testing, allowing it to be done anywhere by anyone at any time. People lack access to affordable, accurate, and rapid diagnostic testing, and that includes millions in the US. The result of this is delayed or misdiagnosis and the spread of disease. In fact, there are over 14 million people that die every year from infectious diseases. This is too many. During the pandemic, testing scaled, but not with PCR or polymerase chain reaction, which is the gold standard and the most accurate and sensitive tests. PCR adoption has not reached its full potential for three main reasons. First, the sample, whether it's saliva or swab that is collected, needs to be purified in order to be amplified to get access to the DNA and RNA. Today, there are toxic chemicals or inhibitors that are used to stabilize those samples that then have to be removed. This requires skilled technicians as well as expensive equipment and reagents. Going the wrong way. Here we go. Thank you. Got it. Okay. So our breakthrough technology is direct to PCR testing. This means you can collect the sample, place it into our buffer, and you can skip the pre-analytic step and go straight to testing or running the sampler. This means that it's faster, cheaper, and highly accurate compared to conventional PCR. In addition, you can simplify the hardware as well as the design. So here you see a picture of a custom-built open PCR instrument, which a third of it is dedicated to the extraction step, running those samples over a column or a bead. That could be eliminated. Below you have an example of the Thermo Kingfisher used for extraction, which also would not be needed. So our estimates are that eliminating these reagents and these toxic chemicals would lower the overall risk to the operators, but also lower the overall operation cost and the maintenance needed to be done in labs. So our technology is built on a clinically validated data set that was proved out during COVID in a CLIA lab in Colorado. We tested over 590,000 patients with SARS-CoV-2, also did testing in flu, and we had 100% concordance with swab compared to conventional PCR. We have an issued patent that is on this extraction-free chemistry. It was issued last July. And this is a patent that is a composition of matter and method, so it covers the chemistry and the workflow. And we have an exclusive license and full freedom to operate with this, so we own the IP, and we have one patent issued, but 5 pending, including on a at-anywhere handheld device that is in proof of concept stage. With our proven PCR direct chemistry, we will be able to do advanced microfluidics and essentially do a lab-on-a-chip technologies, enabling us to do PCR anywhere. Get it out of the lab, simplify it at a low cost. Our target is to get this to be able to do it under $10 on the chip. This is a huge market. It's $20 billion across a number of different segments, and we have large incumbents in this space, including Roche, who I mentioned I worked for before, and have point-of-care devices and also molecular diagnostics instruments in core laboratories. And our goal is to identify these partners, to work with them, and to empower them to simplify PCR testing and to take advantage of what we've discovered and also the proof of concept on the next generation instrument, because there's also players like Visby Medical and Binks who have sexually transmitted infection disease tests that they're trying to sell direct to the consumer market. But again, we think those can be simplified and can be brought down to a price point that makes it more accessible to everyone. The market is 12 billion and that we're going to target. And this is initially in the research and the clinical sample collection media market, molecular PCR, and then point of care, which is growing, we believe, the fastest. We're raising $6 million, and the use of these funds will be to extend our data, increase the number of publications, and also file our 510(k) on the sample collection media and start selling customers the product, as well as licensing to strategic partners and OEMs. And we'll bring on some team to strengthen our execution. We have a plan to build our quality system, publish more studies, as I said, and then our clearance we anticipate will be in mid 2028. On the At Anywhere device, we're going to move that through proof of concept to then prototyping phase, and then start our assay development on not just things like chlamydia and gonorrhea, but also other pathogens that are difficult and an unmet need in areas such as TB, which is of interest to the military and other groups. This is our go-to-market strategy and roadmap. We have our DirectAmp RUO extraction-free buffer that we will start selling in the end of this year. Point of care, we'll start licensing our IP, and then the clinical market will enter that in mid-2028 with the 510(k) approval of the sample collection kit. And then in 2028 and beyond, we will start to partner and license our technologies to molecular diagnostics companies. We have a clear path to break even, both with the reagent business that will help fuel some of our growth, and then with the licensing model. And we have a plan to be capital efficient, which would allow us to have a potential strategic exit in 4 to 5 years. There are 6 or 7 acquisitions that have been done in this space by many of the strategic players you saw there over the last 10 years, ranging anywhere from 140 to over $400 million. So we're optimistic that this is an attractive and growing, interesting market. We have an experienced team. Recently brought on a head of R&D who had experience working at 23andMe and Affymetrix, also a number of different small device companies, including point-of-care devices. Jay Faust is our business development lead, and he has experience licensing technologies both across diagnostics and pharma, including companion diagnostics. We have team members that were at the previous lab in Colorado where we did all the COVID testing, Summit Biolabs. And then the board members have experience in both molecular diagnostics as well as across pharma in leadership roles, including our co-inventor and board chair, Bob Bloomquist. We have an experienced team of advisors and product development experts at both the University of Colorado who have experience in engineering, microbiology, microfluidics, as well as University of Colorado, and then an expert in the development of different devices over the last 20, 30 years, Mickey Ardea. So great team and growing group of advisors. So in conclusion, we are going to deliver affordable, accurate, and rapid diagnostic testing for infectious diseases and PCR. We have a strong group of advisors and team members who are proven in diagnostics, and we have a landmark patent that we're very proud of and 5 pending that are moving through, both in the US, in Europe, and in select countries in Asia. We have 590,000 clinical tests performed. We have a clear path to monetization and to licensing our technology. We're asking for $6 million, and we believe that we will have a sustainable business and also multiple exit pathways. And in addition, we also believe that this is really something that is very much needed in the world. And with more access, we could reduce the number of deaths, not just here in the US, but around the world with PCR at anywhere. So if you're interested, please find me after, and thank you for your time and attention.